Hello, Aman. Uh, Sorry. Okay. Am I audible, guys? Hello, Pratika. Okay. Guys, am I audible? Can you please quickly tell me that whether I'm audible or not? Hi. Hello. Hi. Okay. Uh, okay. So, uh, I think few people already joined. So, I think we should start and then let's see. Okay. Okay, audible. Okay, okay. Thank you. Thank you. Okay. Chalo. So, guys, first of all, one thing that in previous one classes, I just somehow I just mentioned that H3 is the most conserved. I think I that somehow I just confused with some epigenetic marker. That's why actually H4 is the most conserved, and I have received some messages from you guys, but I'm extremely sorry for this mistake. Actually, I have not any intention to do it, but still. Okay. So, hello, Tanushree. Okay. So, guys, I think we should start. Okay. So, our first question is bacterial flagellin is detected by TLR3, TLR7, TLR5, TLR9. Okay. So, bacterial flagellin, we all know bacterial flagellin. Okay. Uh, can you please quickly answer this question? Bacterial flagellin is detected by TLR3, TLR7, TLR5. Quick, guys, please do it, hurry. Uh, First up, guys, first up, I think we have 40 question today. So if you just quickly answer this question, then we can 40 question we have, and all this question is very, very important. I have selected for you guys. So please do it. Okay. So the answer will be TLR5. Yes, TLR5. TLR5 is the correct answer. Okay. Yeah, C. Okay. Chalo, next question. Hello, good morning, Priti. Chalo. Next, which is not antigen presenting shell? Which is not antigen presenting shell? Okay. B shell, T shell, macrophage, and dendritic phage. Okay. We all know that there is some antigen presenting shell. They just present the antigen to the T shell because T shell can't directly attack or can't directly do anything on the antigen. So T cell actually needs something. Okay. B. B. Okay, okay, guys. I have already mentioned the answer. Okay, chalo. Question number 3. Which cell has both myeloid and lymphoid progenitor? Macrophage, dendritic cell, RBC, and B cell. Which cell has both myeloid and lymphoid progenitor? Okay. Hello. Hello, Kes. Hi, Sampad. Hi, Mihir. Okay, everyone is saying it's B. Okay, yes, it's B. Fantastic, guys. One thing I have to do, I have to just off my webcam because I think something going wrong. Okay, then I think it can be happen. Okay, hello. Now, my fourth number question is that so Chediak-Higashi syndrome. Okay, Chediak-Higashi syndrome is due to lack of macrophage, T cell, NK cell, and mast cell and neutrophil. Okay, Chediak-Higashi syndrome. अच्छा, क्वेश्चन पूरा नहीं दिख रहा है। हाँ, ऊपर ऊपर थोड़ा कट हो रहा है, आई कैन सी, बट आई डोंट नो व्हाट्स हाउ टू रेक्टिफाई इट। ओके, I will check tomorrow. Don't worry. I'm just reading the question, so just listen it. Celiac Higashi syndrome is due to lack of macrophage T cell, NK cell, and mast cell. Okay, everyone is saying it's C, NK cell. Fantastic, guys. Fantastic. Chalo, let's move. Okay. Now, question number 5. Which molecule help NK shell? Okay. Which molecule help NK shell to participate in antibody-dependent cell-mediated cytotoxicity? Which molecule help NK cell? We know for NK cell, okay, NK cell needs something, okay, to participate in antibody-dependent cell-mediated cytotoxicity, ADCC. Okay, someone is saying D. Okay, Pratika. Okay, fantastic, fantastic, Aman. Okay, fantastic. Okay, guys, it's D, CD16, because we know only CD16 present in the NK shell. It is present in TC shell. It is present in T H shell. Okay, have to remember. Okay, fantastic, everyone, guys. What is very poor antigen? My question number six is this is question number six, and my question is what is very poor antigen? Okay, nucleic acid, protein, carbohydrate, and can't be said. Okay, there is something very poor antigen. Okay, what is very poor antigen? Nucleic acid, protein, carbohydrate, and can't be said. This is question number six. Do it faster, guys. Okay, Pratika is saying it's A, nucleic acid. Aman is saying it's A. Okay, okay, fantastic, guys. It's nucleic acid. Okay, even protein is the most strongest antigen because due to protein, we know that lot of antigen they just express themselves. Like suppose if there is some kind of viral mRNA like this, they just go inside the host cell and then they make protein, and our immune system can recognize only in this form because there is lot of factor, molecular weight, complexity, everything. Okay, fantastic. Question number 7. Which is not true about MHC molecule? Okay, major histocompatibility complex, MHC molecule. It is polygenic, it is polymorphic, it is dominantly expressed, and it helps to present antigen. Okay, so my question number 7 is which is not true about MHC molecule? Okay, polygenic, polymorphic, dominantly expressed, and help APC to prevent antigen. Okay, do it faster, guys. It's a really fantastic question. Okay, dominantly expressed, Aman is saying. Okay, let's see for other people. Okay, Shatabdi is saying it's C. Okay, do it. Uh, okay, so yeah, Fazal is saying it's C. Okay, fantastic, guys. It's dominantly expressed. It is not true because MIC is codominant. Okay, yeah. Hi Sneha. Yeah, it's codominant. Question number 8. Okay, each light chain of antibody. Okay, each light chain of antibody contain uh, few amino acid, and heavy chain contain how many amino acid? My question is that we all know that this is the thing. So this is the light chain and this is the heavy chain. So my question is that, yeah, let's see. Yeah, it's C. Okay. So my question is that, yeah, can you please tell me that light chain how many amino acid and heavy chain how many amino acid? It's really good question. I think this question has come in some different form in any year. Okay. Okay. 64. Okay, Fuzia. Okay, fantastic. Preeti. Okay. Nice, guys. I'm really pleased to see it. 220 and 440. Yes. Yes, Fazor. Yes. Okay. Chalo. Question number 9. Okay. If you perform papain digestion of immunoglobulin G, okay, you are performing papain digestion of immunoglobulin G, then how many band you deserve in SDS page? Okay, you are performing IgG papain digestion. I hope you know What is the meaning of papain digestion? You have to know the meaning of papain digestion. Okay. Can you please quickly tell me? 2 Fab and 1 FC. Yes. But how many bands? How many bands you can see? Fazal is saying 3, Swatabdi is saying 2, Mir is saying 3, Pooja is saying, no, I am not asking about the fragment, I'm asking about the band. Sampad is saying 3. Okay, guys, it's 2. Because what happened in case of SDS page, as the 2 Fab fragment, okay, the 2 Fab fragment, their molecular weight is same, so you can't really differentiate between them. They will be a same band, okay, their intensity will be more, and one band is here and one band is here, like this. So this is for suppose FC1 and one is for Fab. There's 2 band, okay. Next. How many CDRs are there in IgM? Fabulous, fantastic question. How many CDR? Okay, complementary determining region. I hope you can remember this. This is the antibody. There is something called present in both heavy chain and light chain. Okay, complementary determining region. So 2. Yes, yeah, sample 2. 2 fabs give 1 and 1 FC. 2. Now just do it faster, guys. How many CDR, okay, are there in IgM? Priti is saying 60. Okay, Pratika is saying 60. Fantastic. Fazal is saying 12. Okay, Satat is saying 60. Yes, 60. Because we know there is in one chain, okay, one chain there is 3 CDR. Just remember, guys. So in one antibody, if there is IgG, then what will happen? It is 12 CDR. But as IgM, you can write equivalent to 5 IgG. So that means it's 60 CDR. Okay. So you have to put here 60. Yes, fantastic. Now question number 11. Okay. Uh, Which antibody has highest half-life? My question is, which antibody has highest half-life? IgG, IgA, IgD, IgE. Okay. Which antibody has highest half-life? Yes, Fazal. 5 into 12. Yes, fantastic. Do it faster. There's a lot of question from immunology and microbiology. This is virology. Okay, you are saying A. Okay, Shampoo is saying A, Priti is saying A, Pooja is saying A. Okay, everyone is saying A. Shoma is saying A. Yes. Okay, yeah, it's A, IgG. Okay, Mary is saying A. Okay, chalo. Next, the first signal of B cell activation. Okay, I hope you can remember the activation of B cell signaling. Okay, B cell and something called like this. Okay, this is B cell, this is T cell, T helper cell. B cell need this help. The first thing is that MIG, okay, MIG to AG interaction. That means membrane bound immunoglobulin to antigen interaction. The first and there is something like CD4, CD40 in B cell, suppose 40 ligand, and the CD40L cytokine, and BCR there is cytokine receptor. It secretes cytokine. So can you please quickly tell me from which things? Okay, sorry, I have already just click here. Okay, this the first signal comes from, yeah, A, because at first to do something, okay, your the antigen will bind here to the membrane bound antibody. Okay, no, it's A, guys. The first signal I am asking about, the first signal uh, of B cell that comes from when antigen bound to the receptor, B cell receptor, the first signal then comes. Then when it is the first signal, it is the second signal, CD40 and CD40L. It is the second signal, and that it is the third signal. Okay, remember the first signal is that, second signal is that, and third signal is cytokine BCR receptor interaction. Okay, chalo, let's go. I don't know why the screen is just quite short. Okay, now question number 13. Okay, in two sorry, kya hai bhai? Oh, sorry, I also cannot read. In secondary response, which antibody is predominant? Okay, in secondary response, which antibody is predominant? IgG, IgM, IgT, IgE. Faster. I think it need only just 10 seconds. to type. In secondary response, which antibody is predominant? Shoma is saying C, Priti is saying A, Shampad is saying C. Shampad, you have to read more. Pratika is saying A. Okay, okay, yes, guys, A, IgG. Because we know in case of primary immune response, IgM is quite high, but still, in case of secondary, if it is IgM, okay, it is IgG. Okay, so in secondary immune response, IgG just play a beautiful role. Okay, okay, chalo. Uh, in monoclonal antibody production, my question is that in monoclonal antibody production technology at hat medium, okay, at hat medium, which cell will survive? Okay, so I hope you know the hat medium and HGPRT, all these things, because you have to know. Okay, in monoclonal antibody production technology at hat medium, which cell will survive? Okay, B plus M fused, B plus M fused. Okay, chalo, B plus M fused. Because what happened? Because B cell will not survive because it is mortal. Okay, for long, and it is HGPRT minus, HGPRT minus. Okay, so that's why only B plus M cell, because it has both property. It can synthesize. Okay, there is HGPRT also, plus there is something like immortality. Okay, so it can survive. Yes, chalo. Question number 15. IgG1 and IgG2 are known as allotype, isotype, idiotype, and isotonic. I don't know really what's this, but you have to tell me. IgG1 and IgG2 are known as, okay, uh, IgG1, this is IgG1 and this is IgG2, okay, are known as allotype. I hope you know the meaning of allotype, okay, and isotype and idiotype and isotonic. So my saying A, allotype. Okay, let's see. Nidhi is saying A, Shampad is saying A, everyone is saying A, so it's A. Okay, and what is isotype? Like if we take IgA, IgD, IgE, then they are called isotype. And idiotype, suppose your own IgA is responsible to antigen X and your own IgA is responsible to antigen Y, but they are quite different. Then the difference come, and allotype. No, isotype is like this: IgA, IgD, IgE. Okay, yeah, this is the isotype. That's why we know the isotype switching. There is a terminology, isotype switching. Okay, rag recombinase, this is rag recombinase. Okay, rag recombinase help in, this is MSQ, typical MSQ. It is rag recombinase, it is an enzyme. It help in BCR diversity, TCR diversity. Okay, only soluble IgG diversity and MHC diversity. Okay, the question is rag recombinase help in BCR diversity, TCR diversity, only soluble IgG diversity, and MHC diversity. Quick, guys. B and A. Fazul is saying B and A. Let's come on. Shama is saying A, B, D. Okay, okay. Shampad is saying sorry A and V. Okay. Here is Pratika and Vijaya, Aman, A, B, D. Okay. नीति is saying A, B, D. Fantastic. चलो देखते हैं पहले और कौन कौन, कोई तो कुछ डिफरेंट बोलो, लाइक A, B, C, D और B, C और C, D. The correct answer will be A and B. Really, MHC diversity, there is no role like rag recombinase. Okay, no rag. There is no role of rag recombinase in MHC diversity. Okay, so only B cell diversity and T cell diversity, rag recombinase just make a role. Okay, don't know, sir. Okay, sorry, not an issue. Question number 17. Okay, which enzyme is responsible for somatic hypermutation? Which enzyme? Okay, the question is which enzyme is responsible for somatic hypermutation? CID, AID, VID, UID. Recombinase role in VDJ. Yeah, Fazal, Reagar recombinase role in VTJ recombination. So there is also T cell receptor. It has to maintain TCR also, but MHC, in case of MHC, it does not play any role. Okay, so just quick, which enzyme is responsible for somatic hypermutation? Okay, someone is saying still C. Rupal is saying C. VID, भाई VID कौन एंजाइम होता है? VID, Priti is saying C, Fazor is saying B. Okay, the only good guy is Fazor. Okay, it's AID. AID only play a role in somatic hypermutation. Nothing like VID, CID, UID. Okay, अरे भाई I really don't know. ये तो I really don't know them. But see, what is CID? You can say that uh, criminal investigation department. These kind of things. Okay, chalo. Uh, cytokine. My 18th question is that cytokine shows which property? Okay, adenosine deaminase. Yeah, you have to just check adenosine deaminase maybe. Okay, cytokine shows which property? Okay, it's also MHQ. Okay, अच्छा. Activation induced deaminase. Yes, activation induced deaminase. अरे full form पे पीछे मत पढ़ो भाई seriously. You just like D for deaminase, you can get it. And you just know, you have to know just little bit mechanism, okay, and then you can easily get it. Chalo, question number 18, cytokine shows which property? Okay, guys, my humble request to all of you, don't be too clever, okay, in your exam or during the preparation, the last 5 days or 4 days, कि हर एक फुल फॉर्म देखना चाहिए, ऐसा कुछ नहीं, ठीक है, just recall it. Okay. Cytokine shows which property? MSQ. It is okay. Quick. Pleiotrophy, redundancy, synergy, antagonism. And antagonism. Okay. There is four property: pleiotrophy, redundancy, synergy, and antagonism. Cytokine. I hope you know the cytokine. There is lot of interleukin, interferon, chemokine, cytokine, all these things, na? Like tumor necrosis factor is there, cancer role, interleukin 6, interleukin 2, lot of things, na? Guys, all will be happen. Like they all antagonist. Sampath, why you're not saying? And it's also antagonist. Suppose one cytokine is just doing one thing. I think there is IL-2 and IL-4, maybe. I'm not sure. Suppose, well, let chalo. Suppose IL-2 just activate macrophage, okay, and IL-4 will inhibit it, okay. This kind of things, behavior also shown by cytokine. Yeah. Yeah. So question number 19, which hypersensitivity is mediated by T cell? I hope you know the hypersensitivity means that your body is really hypersensitive to any kind of things. We generally know in the form of allergy, whatever you call it, allergy, so-called like some kind flower allergy, gluten allergy, these kind of things. Hmm. Okay. Someone is saying C. Someone is saying C. Priti Sharma Fujia is saying D. Okay. Aman is saying D. Yes, it's D. Okay. D is TH mediated. Okay. T cell mediated, and it's delayed. Okay. And it's delayed. Remember, it's delayed hypersensitivity. Like if something happened, you can get after 20 days or 10 days. This is due to the type 4 hypersensitivity. Okay. And type 1 is very quick, very fast. Suppose egg allergy, yeah, type 1 is very fast. Okay. Chalo, question number 20. Which of the following is recombinant DNA vaccine? And it is our last question of immunology, but then we will start microbiology. Okay. Which of the following is recombinant DNA vaccine? Okay. Sampath, it's immediate. Okay. There's hepatitis C, hepatitis B, smallpox, and influenza. There are four vaccine. Okay. And guys, please quite SARS-CoV-2, just observe few vaccine, okay, few vaccine, their type, this kind of thing. Okay, because they will ask you, they can ask you about something what's going on, because you are all time hearing SARS-CoV-2, SARS-CoV-2, but you have to know which vaccine is based on which property. Okay, so please take care it. Which of the following is recombinant DNA vaccine? Can you please quickly tell me? This is hepatitis B, C, smallpox, influenza. Which one? Yes, B. Yes, hepatitis B. It's that thing. Okay. Actually, there is no effective vaccine against hepatitis C. Like they are trying to prepare it. Okay, and there is a lot of vaccine against influenza A. Okay. Guys, can you please quickly tell me which of the following uh, virus show the property of antigenic shift? Which of the following virus show the property of antigenic shift? Quickly tell me. It's a really fantastic question. I hope there is a fourth thing: hepatitis B, C, smallpox, and influenza. Antigenic shift. I hope you may know the meaning of antigenic shift, because in case of seasonal things, like when season change, so now time now it's the time. Okay, so you have to be prepared for the attacking of influenza. Yes, influenza, influenza virus, so antigenic property. Okay, guys, please stay tuned. Only 1 minute. I just want to off my camera because I think for this there is some issue. Okay, uh, just 1 minute. Just let me check. Okay, guys, so this is, yeah, now this has gone. I think so, but still there is something. What is this? I don't know. Okay, okay, chalo. This is our question number 20, and then it's our question number 21. Okay, so now microbiology start. Okay, so how RNA virus replicate into the body? Please, please just think about it. Don't leave, just think about it. How RNA virus replicate into the host body? They use host replication system. They integrate with host genome. They carry their own replication enzyme, and none. The question is, how DNA virus replicate into host body? Okay, I am reading the question because I think there is something problem. Okay, how DNA virus replicate into the host? It is MCQ, guys, not MSQ. So sample क्वेश्चन काट हो रहा है सर. हाँ हाँ, आई नो क्वेश्चन कुछ प्रॉब्लम आ रहा है, बस प्लीज लिसन टू माय वर्ड्स. आई एम जस्ट mentioning the whole question. I am reading this question. Okay, how RNA virus replicate into the host body? Option is they use host replication system, they integrate in with host genome, they carry their own replication enzyme, and none. Okay, so my saying C. Well, guys, please faster, faster. Preeti, sir, DNA virus. I am mentioning RNA virus. भाई लिखा है RNA virus. I am reading also. Okay, some Fazol is saying A. A क्या? No. अरे मेरा गलती से टिक हो गया. It's C. They carry their own replication enzyme. They can't use host replication system because in our host there is nothing like RNA dependent RNA polymerase. For replication of RNA virus, what need? It need RNA dependent, okay, RNA dependent RNA polymerase. It needs RNA dependent RNA polymerase, but we don't have RNA dependent RNA polymerase, so they have to enter our system with their own enzyme RNA dependent RNA polymerase for their proliferation. For DNA virus, it will be A and B. Sampad, my humble request to you, please don't be clever, okay? Just think whatever is given. If you don't change this habit, what happen in exam? You will also suffer. So please, I'm not blaming you. I'm just saying you the fact. Question number 22. Which virus has gapped double-stranded DNA genome? Which virus has gapped double-stranded DNA genome? There is hepatitis C, adenoviridae, flaviviridae, hepatitis B virus. Okay. Which virus has gapped double-stranded DNA genome? Okay. mRNA independent. Paul. Paul. Dekho, one thing is that Shema, the thing, please don't always think if virus has SS positive stranded RNA, it or minus SS RNA, it is not indicating always that it is mRNA. It is really a misconception of us. It is not, really not. After your exam is over, please check on YouTube lecture series of Professor Vincent V. Uh, Rasinello. Professor Vincent V. Rasinello, and there's one of his book is Principle of Virology, I think so. Okay, so which virus has gap DNA? It is hepatitis B virus. Yes, it is hepatitis B virus because you can see here that something is incomplete. Okay, the double-stranded DNA genome is not complete. Okay, in case of gapped DNA genome. Now, question number 23. Okay, so there is a mathematical question. You have to calculate it and tell me just what can be happened. Okay, so there's a viral stock, and viral stock has 10 ml. You are just putting 0.1 ml. This is 0.1. This is 0.1. Everything. Okay, you have put here 0.1 ml to here, and then 0.1 ml here, then simultaneously. Okay. Now the remaining is 9.9. It's maybe water. Okay, so add this add this step. Okay, can you please quickly tell me how many PFU you can observe? Okay, how many PFU you can observe? There is there is 2 into 10 to the power 8 cell. Question not visible. Question not visible, Shama, but I think question is visible to me because मेरे पास भी मेरा YouTube on है in my phone, that I can check that whether it's any issue or not. Guys, question is not visible kya? I think it's okay now. Please try. It is the main concentration, okay, of main virus, and you are doing 1, 2, 3, 4. Okay, just write down 1, 2, 3, and 4. So you are doing, okay, 10 to the power minus 4 dilution. So it is your dilution factor. So just yes, the above portion of the question is cut. Ha, Sampad, I know the above portion is cut. So I think I will check it tomorrow. Don't worry. But I am reading the question now. Yeah, just so the concentration in the stock is 2 into 10 to the power 8, and okay, and you are putting you are taking, okay, 10 to the power minus 4 as a dilution. Okay. Therefore, I am not asking you about that I have taken 0.1. I have not taken 0.1 and then I am calculating. No, I am not doing it. I am directly calculating the whole virus, whatever is present here. Okay. So I am not doing this. Okay. So the thing you will get 2 into 10 to the power 4. Always just keep your mind open that whatever is asked in the question, how many plugs can be seen here. I am not asking that I have taken 0.4, 0.1 ml, like whatever you have done, like in case of black acid. But I am randomly just I want to ask you this. Okay, so you have to just whatever you have diluted, okay, from the concentration, you have to multiply the dilution factor, nothing more than this. Okay, chalo. Question number 24. Okay, 2 into 10 to the power 4. Yes, yes. What is basic difference between bacterial growth curve and virus growth curve? It is really, guys, really very important to know it. Okay, what is the basic difference between bacterial growth curve and viral growth curve? Virus curve has more slope. Okay, so this is and the question I just want to ask you, that what is the basic difference between virus growth curve and bacterial growth curve? Many people will ask this question in any interview and as well as JAM exam also. So viral curve has more slope. Bacterial curve never reach declining phase. Okay. Viral curve initially show no growth. Okay. Bacterial growth curve is sigmoidal, but viral growth curve is J-shaped. Okay. Quickly tell me. So I'm asking you that what is the basic difference between virus growth curve and bacterial growth curve? Just think once that what is there. So question D. Okay. So someone is saying D, sigmoidal and J-shape. Fazol is saying D. Everyone is copying. Ha ha ha. Oh no 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 Preeti. RFI. No guys, it's not D. It's not D. Remember, it's C. Okay. What happened? Initially, virus show no growth initially because they have to penetrate our cell. They have to make their own whole system. They will make it, and at that time they are busy. So they don't rupture the cell. They don't burst the cell. They don't kill the cell. They just do whatever they want to do, and then they will leave. Yes, Aman, it's C. Viral curve initially show no growth. If you just show virus growth curve very carefully, you can show, you can see that this kind of curve, there is no growth initially. Okay, and suddenly when their every system is everything is ready, they just burst up. Okay, हाँ मेरा काम खत्म. Now I will go. No one can just stop me. Okay, the virus just so this kind of graph, whereas bacteria, so this kind of graph. Okay. So remember it. It is one step growth curve. Okay. My question number 25 is the energy for rotating flagella in bacteria is the energy. Yeah, they hijack. The perfect word. They hijack. Wonderful. The energy for rotating flagella in bacteria is PMF, proton motive force, H plus ATPase, F0, F1 type pump, no need of energy. Preeti is saying B, Preeti is in hurry, I hope so. Maybe उसका आंसर सही भी हो सकता है, चलो देखते हैं. Aman is saying B, okay, H plus ATPase pump, okay, everyone is saying B. Pratyush Kumar Datta is saying B. Guys, can you please just once look whatever pump is present here, is there really some kind of H plus ATPase pump, or only the PMF is responsible for the flagellar movement? Just check it once after the completion of the class. As far as I can remember, there is no such kind of pump, so please confirm it. I don't want to conclude that it's PMF only, because as far as I can remember, there is no. Yeah, sure, yeah, Soma, yeah, and I think it may be PMF, proton motive force, because there is no such kind of H plus ATPase pump. Okay, okay. Question number 26. Chalo. In gram staining, okay, which is used as mordant? सर प्रोटॉन मोटिव तो H प्लस ही होगा ना. हाँ भाई, प्रोटॉन मोटिव H प्लस ही होगा, but proton motive cannot do the function through the pump. It can, I think there is some motor protein, okay, mot A, mot B inside the flagella system, so it just make it rotate. There's some mechanism behind it. Yeah, energy is utilized. Okay, okay, proton gradient को H plus ATPase नहीं बोल सकते. हाँ, proton gradient, we can directly say that where there is proton gradient, the H plus act. But I have to check. देखो H plus pump is there, but whether H plus pump is responsible for this or not, It is really not clear. Okay. Okay, I will check it once and then I will inform you. Yeah, flagellin protein. No, flagellin of protein is not like it just formed the flagella. Okay, just then there is a lot of thing na like mot A, mot B, the whole the motor machinery. Okay, I think it may be possible that there is interaction between them, and I will check once. Okay, don't worry, I will check once. Yes. Okay, just focus on this question. In gram staining, which is used as mordant? Safranin, alcohol, iodine, crystal violet. Okay, everyone is saying it's iodine. Fantastic. Now, question number 27. Okay, if multiplicity of infection is 5, I am mentioning that multiplicity of infection is 5. I have mentioned my equals to 5. Yeah, please check because yeah, I also just it's possible that I forgot maybe na. Okay, so the multiplicity of infection is 5. How many cells in a cell culture will remain uninfected? Suppose I'm giving you a cell line HEK253 or something else. Okay, so how many cells will remain uninfected? Quickly tell me, guys. That's really fantastic question. What is the formula that how many percentage of cell remain uninfected? Okay, cell remain uninfected. Okay, equals to e to the power minus of MOI. Okay, just do it, calculate it. Okay, what is the e to the power minus 5? Can you please quickly tell me? No, no, no. Just no, 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 Soma. The thing is not that so easy. So there is a formula, okay, that if multiplicity is x, so e to the power minus x, okay, e to the power minus x amount of cell will remain uninfected. Okay, quickly just do the calculation e to the power minus 5. Then just tell me what is coming. Uh, okay, and when if someone asks you how many percentage of cell is infected, just do it 1 minus this thing. Guys, you're doing or not? Value of E array 5. Just put it in your 1.6. No, guys, like it is really not possible. Minus 5. I think into 100. 0.67. Yeah, the thing is coming that into 100% as for percentage into 100%, so it's coming 0.67%. So 0.67% cell will remain uninfected. Yes, but you have to just uh, multiply it with 100, okay, to get the exact value, and the remaining 99.33% cell infected. Just think once that if you are adding virus to a cell culture, it does not mean that your all cell will be affected. Okay, so and every cell will not get uniform virus. A cell will get suppose 10 virus, a cell will get 2 virus, one cell will not get any virus. So that's how some cell will remain uninfected, and this is the formula e to the power minus of multiplicity of infection through which we can calculate how many cells remain uninfected. Now question number 28. How can, sorry, you can find dipicolonic acid. I hope you have heard the term dipicolonic acid. Okay, you can find dipicolonic acid in, it is typical MSQ question, that is Mycobacterium, Pseudomonas, Bacillus and Clostridium. You can find dipicolonic acid in Mycobacterium, Pseudomonas, Bacillus, and Clostridium. How to do this? Okay, how to do this? Just press in your virtual calculator. I have to check it once, but in your scientific calculator, shift and ln. Okay, shift and ln. A and B. So Shoma is saying it's Mycobacterium and Pseudomonas, Bacillus and Clostridium. Okay, CD. Okay, those guys were saying CD. It's right. Bacillus and Clostridium. They are forming endospore. Yes, and dipiclinic acid is found in endospore. Okay. Question number 29. Nitrosomonas is an example. Nitrosomonas is an example of, okay, chemo-organoheterotroph. I hope you just remember the EEC system. Okay, so Nitrosomonas is a chemo-organoheterotroph, chemolithoautotroph, photolithoautotroph, chemolithoheterotroph. Sampad is saying it's B and C. B and C. It's not MSQ Sampad. Nitrosomonas. I have provided here Nitrosomonas. Nitrosomonas is an example of chemo-organoheterotroph, chemolithoautotroph, photolithoautotroph, chemolithoheterotroph. Someone is so much saying D. Somewhere D. How? Chemolithoheterotroph. Okay. Pooja is saying it is B. Okay. Aman is saying it's B. Okay. Preeti is saying it's B. Okay. Fantastic. Fantastic people. It's B. Chemolithoautotroph. Okay. I hope you know that chemo, litho, and autotroph means they can produce their own food. Yeah, it's a source of energy, and it's the ionic source. Okay. You know these things. That was previous question. Okay. Okay. Sure. 30. Which bacteria can form gas vacuole? My question is, which bacteria can form gas vacuole? Yersinia, Halobacterium, Pseudomonas, Bacillus. Which bacteria can form gas vacuole? Yes, Yersinia, Halobacterium, Pseudomonas, and Bacillus. Yes, do it faster. I don't know. The question is cut. Question is that which bacteria can form gas vacuole? I'm writing here it's gas vacuole. Okay. Someone is saying D. Okay. Preeti is saying D. Fazor is saying B. Okay. Sampad and Aman and the remaining people. I'm waiting for you quickly. Preeti is saying B. Okay. Chalo. It's B. Halobacterium is B. Chalo. Fantastic. Okay. The first discovered human pathogenic virus. Okay. The first discovered human pathogenic virus is polio, FMD. Okay. No, so, but the previous answer is I think it's Halobacterium. Yersinia is not. It's only Halobacterium. Okay. The first discovered human pathogenic virus is polio virus, FMD, yellow fever, pox virus. Okay. Hazard is saying FMD. Okay. Someone is saying it's D. Someone is saying it's B. Okay. Chalo. Let's see. Someone is saying B. It's very important question, guys. Very important. I am putting here. No one is saying the correct answer. How? How this is possible? You people are really blind. Don't mind. Sampad, you too. Preeti too. Okay. It is D saying D. Pox virus. Okay. Kyun? Because it was discovered by Edward Jenner. It's yellow fever. Remember, it's yellow fever. If FMD will only happen, okay, if there is, I ask you, animal virus. If the question is like that, what is the first discovered animal virus, then we only can answer that it is the FMD, foot and mouth disease. Okay, please कृपया करके थोड़ा ध्यान दो. Otherwise, you will do the same mistake whatever I have done. Okay. Question number 32. Which virus has segmented RNA genome? This is wonderful question, guys. Really wonderful. Which virus has segmented RNA genome? Because from microbiology, no one is going to ask you some hyphanda. They will ask you something like this, I think. So SARS-CoV-2, influenza, pox, and hepatitis, and rabies. Okay. Which virus has segmented RNA genome? The question is which virus has segmented RNA genome? I hope you know the segmented meaning, like suppose this kind of thing, not integration. Okay, but in what will happen in case of virus, there is some integration. Okay, but few virus have segmented. SARS-CoV-2, अरे वाह 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 बढ़िया, feather saying P, influenza, quick 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 guys. It's obviously influenza. Okay, it is segmented genome. SARS-CoV-2, it's only plus ssRNA. I hope now. Yeah, it's plus ssRNA. No, pox is DNA. Segmented genome? No, pox is not segmented. You can't say that pox is segmented. Question number 33. Pure culture was first prepared by Pasteur, Von Smith, Koch, and Harold Vermas. Okay. Pure culture was first produced by Pasteur, Von Smith, Koch, and Harold Vermas. This is good question because I think I received this question get 2020. I hope so. This kind of thing get 2020. Okay. Every people is saying C. Okay. Robert Koch, pure culture. Okay. There is Koch postulate. Please read it once. They will just give you one like 4 option. Then they may ask you what is not belong to the Koch postulate. Okay. And there is something like river postulate. Okay. River postulate. Please just look once. River postulate. Koch postulate and river postulate. Okay. Now, question number 34. Okay. E. coli generation time is 20 minutes. I am mentioning that E. coli generation time is 30 minutes. Sorry, 20 minutes. In lab, if you start with 2 into 10 to the power 3 E. coli after 2 hours, okay, how many E. coli you are getting, like you are expecting to get. So your time is 2 hours, and generation time, that means number of generation. How you will calculate how many generation happen? So just 2 hours means 120 minutes, and generation time is given 20. That means 6 generation. Okay, you know that nt equals to n0 into 2 to the power n. Okay, simply apply this 2 into 10 to the power 3 into 2 to the power 6. I hope you will get 64 into 128 into 10 to the power 3. Yes, Sampath, 128 into 10 to the power 3. Yes, learn to lead किसका है भाई पता नहीं. चलो, question number 35. So Clostridium, it is Clostridium written here. Please don't think that ये कुछ अलग चीज है. Clostridium is a facultative anaerobic, obligate anaerobic, facultative aerobic, and microaerophilic. Okay, Clostridium is a obligate aerobic, obligate anaerobic, microaerophilic, and facultative anaerobic. Do it. Clostridium is a facultative anaerobic, obligate anaerobic. Like you know the meaning of now, like facultative anaerobic, like they need, they can survive in the presence of oxygen, but if you apply oxygen, then there is nothing such kind of thing. They can. Obligate means they need. Facultative aerobic, that means they can grow better in presence of oxygen, but if you put them in absence of oxygen, they can grow. And microaerophilic means the environment is quite tough, like that Treponema pallidum, okay, this kind of things. So the microaerophilic environment. Okay, Sampad is saying it's A and facultative anaerobic. No, Clostridium is obligate anaerobic. Clostridium is obligate anaerobic. Okay, it can't grow in the presence of oxygen. Remember. Yes. Now, question number 36. Okay, which is not true about archaea? I am asking you about archaea, nothing more than that. They grow in extreme environment, okay, and they uh, start translation. They start translation using FMET. I hope you know the meaning of FMET, formyl methionine, and they are sensitive to uh, anisomycin. So they are sensitive to anisomycin. It's also an inhibitor of protein synthesis, and they don't have nucleus. There is something, okay, in case of archaea, or we can say archaea, whatever. Mihir is saying it's B. Okay, true बताना है ना? हाँ, not true. Not true बताना है भाई. Which one is not true? Okay, like extreme environment, not only. D. Soma is saying D. They don't have nucleus. It's not true. So Soma, according to you, they have nucleus. Okay, I am citation, giving the citation of your name, whatever. Okay, B not true. Yes, B is not true because they always initiate their translation through methionine, like in case of eukaryote. Okay, they just begin their translation through methionine, not fmet. This is the main difference between archaea and bacteria. Please remember. No, no, Fuzia, not D, not D. It is true. They don't have nucleus. It's true. I am asking you about which one is not true. Okay, they don't have nucleus. Really, they don't have nucleus. Fuzia is repeatedly saying D, D. I don't know. Okay, sorry, sir. Okay, chalo. Hmm. To 37. Ciprofloxacin. The here is written it as like ciprofloxacin. Okay, ciprofloxacin inhibit DNA polymerase, RNA polymerase, DNA repair. Okay, DNA gyrase and ORF misreading. Ciprofloxacin is an antibiotic. Okay, RNA polymerase, DNA repair, DNA gyrase, and open reading frame. Quick, don't. अच्छा 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 don't. Okay, so everyone is saying C, C, C. Fazar is Priti is saying C. Sorry, Sampad, Aman, and Priti, the remaining people. Okay, the option is C. Okay, DNA gyrase. Okay, so ciprofloxacin inhibit DNA gyrase. Now, question number 38. Which of the following inhibit directly bacterial metabolism? Fantastic question, guys. This type of simple question you have to practice, not in depth. Okay, so which of the following inhibit directly bacterial metabolism? Okay, DNA gyrase. Yeah, anything you can say. So which of the following inhibit directly bacterial metabolism? Okay, there is something they are not interfering in any kind of DNA, RNA, protein synthesis. They directly interfere with the metabolism. Like it may be amino acid metabolism, it may be nucleic acid metabolism, this kind of thing. Yes. Huh? Okay, everyone is saying C, 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 C. Somebody is saying C. So what is the function of dapsone then? Then what is the function of dapsone? Pooja is saying C. Everyone is saying C. Oh my god. Mitomycin C. It's a cross-linking agent. I hope mitomycin C, Sorlon, they're cross-linking agent, like in case of DNA. Can you please quickly tell me, so what is the function of dapsone, isoniazid, dapsone? What their function? Velomycin cuts the DNA. I'm not asking about velomycin. I'm asking about the dapsone. Okay, dapsone directly interfere. Okay, dapsone is the only thing that directly interferes with the metabolism. Dapsone is there, isoniazid is there. Please, guys, read once. Isoniazid used in TB. Please read once the whole uh, antibiotic list. There is the antibiotic list. You can get it. I think Prescott and as well as Pranab Kumar, you can get it. So please read once the whole thing. Which antibiotic is used in which case? So what is particular function? Okay, नहीं पता. Dapsone. So dapsone just interfere with the dihydrofolate reductase mediated metabolism. Okay, please check it once. 19, sorry, 39. Lambda phage have, wonderful question, double stranded DNA, single stranded RNA, double stranded RNA, and single stranded DNA. Lambda phage, lambda. Hope you remember lambda phage. I am just waiting for your answer, and then we have only one question. Okay, we are in time. Okay, chalo, lambda phage. It is given, it is lambda. Okay, lambda phage is double stranded, single stranded, double stranded RNA, single stranded DNA. Okay, someone is saying D, someone is saying D, Sampath is saying D, Sampath, everyone is saying D. Acha. So can you please quickly write some single stranded, some double stranded DNA name? Okay, can you please quickly give me some example of double stranded DNA example? I just want the example. Please type here. I'm waiting for your answer. Just quickly type here, and then we have only one question. I want you to type here double stranded DNA name example. Okay, quick, quick. It's very easy. I hope you don't need some extra time. Uh, T4. Okay. The other people could please do it faster. Where is lambda? Okay. T even T odd. Okay. Now, okay. Uh, is there any SS phage virus? Is there any SS phage virus? Please remember these things. Is there any SS phage virus? Okay, ssDNA 5x174. Okay, acha. So why you are typing here lambda phage as an SS? So why you are typing here lambda phage as an SS? And Aman, you was mentioning the T even T odd. So I hope you know it. T even T odd lambda. They both belongs to dsDNA, double stranded DNA. Yes, double stranded DNA lambda. That's why I'm repeatedly asking you the whole thing. Please remember all the fudge, all the fudge. And when you will forget, just remember where there is T even, T odd, then there is lambda. Okay, so the answer will be dsDNA. Okay, we have one more question, and it is the last question today, which is not known as oncovirus till now. Why I am writing here till now? Because we really don't know more about virus. Suppose you are thinking that okay, SARS-CoV is not an uh, oncogenic virus, but tomorrow it will be discovered, yeah, SARS-CoV causing cancer, SARS-CoV getting integrated, then they are doing these things. That's why. Dengue, okay. Someone is saying dengue. Someone is saying B, EBV. It is Epstein-Barr virus. It is Rouse sarcoma virus. Sampath is saying A. Yeah, it is dengue. Dengue is still not known as oncogenic virus, but all the three is known as oncogenic virus. Okay. Guys, tell me one thing. Have you found this today's session as helpful session for your preparation? because I really don't want to just spoil your time. It's very important for you, and it's your important moment. Okay, so tomorrow, if like today I have a little bit fever, so I can take one season more. Otherwise, it will be better that if I take one season more. So tomorrow we will take, I think genetics is there and evolution is there. They are both important. Population genetics, please कृपया करके थोड़ा ध्यान दे. Population genetics is very important. Okay, and yes, अच्छा, really good. Okay, thank you, Shampal. So population genetics, genetics and evolution, just simple concept याद करना बस. And then if we have time, we will do plant physiology, animal physiology, and ecology another day. Okay, animal and plant physiology. Okay, plant physiology we will do, I think what can we, like day after today, like tomorrow, Tuesday, I hope so, Tuesday. But guys, please just cooperate with me, because I don't think so that I can do it in at around 10 p.m. So I somehow I have to change some time to modify it. So I will ask you in group. Please respond me as soon as possible so that I can just schedule it that I have to take this class at at that time, because everything is not in our hands. So we have some works in our lab and we have coursework, so we have to finish it according to it. We have to plan it, and I have to ask my mentor that whether I have work at that time or not. So please just quickly respond, and then I will ask my mentor, and then I will schedule class for you. Okay, and I hope you need it. Okay, chalo, bye bye.